Penny, you are ready?
Ready.
Where are you from?
I'm from the UK.
From the UK.
Still united.
Still united. So you are not a Scottish guy?
No, I'm not Scottish. No.
But you need the Scottish money.
They're oil and gas.
Very good. So, start your talk.
Okay, good afternoon everyone. So yeah, as was said, I'm Paddy. I'm here to tell you why I'm a convert. So I work in the BIG-NSE group and do work on biosynthesis. So there we go. So I'm going to tell you today how you go from some yogurt through to making new medicines, new fuels, even new materials, making some polymers. A little bit about me first of all. So I'm a synthetic organic chemist. I'm a dyed-in-the-wool, real hardcore synthesis person, so this sounds a bit weird that I'm going on about biocatalysis. So for my masters, I was doing biomimetic microbial synthesis. These I was basically copying nature. So they make all these compounds, these top ones here, so the top three, natural compounds, they look like drug molecules, really interesting stuff. Then for my doctorate, I also did some more natural product synthesis. This time not biomimetic, I was using catalysis and heavy metals, all this kind of stuff. To make these two compounds at the bottom, which are both natural products. So this got me thinking, okay, nature's obviously making these for a reason, they're quite complex. And nature kind of does it a lot better than we do. So it starts with amino acids, so this is lysine on the left, and with a bit of magic, it goes through to these natural products really, really quickly, like within a matter of days. It took me several months to produce the compounds, even after I just done the whole synthesis. So yeah, nature's better at this than me. So this tree here on the left, you probably know as German Goldbeet. It's actually called the laburnum tree. And it's really toxic, full of all these toxic alkaline compounds at the top. But they're actually really useful for medicine. So we can take these as starting molecules for treatments for Alzheimer's, Huntington's chorea, lots of diseases of the brain that can treat with these types of compounds. And we also use them as a natural product directly. So the one on the right is called cytosine, and this is actually used to help people give up smoking. So it's also got some really simple uses too. But I'm here to tell you about why we use biocatalysis. Because we can start from something like yeast or bacteria, just grow a culture of them, and they can make compounds for us. So, like, you know, penicillin cures gonorrhea in 4 hours. Wow. We can also make beer. Everyone loves beer. It's been around for about 6,000 years, along with bread and cheese, things like that. We can also then make plant extracts. So, the one on the top right corners for artemisinin, which is a treatment for malaria, and it's grown from a load of plants in China. We extract all the compounds out, or we can use biocatalysis to do this as well. We can also make human compounds, so like testosterone or EPO, which was used by Lance Armstrong quite a while, winning quite a bit of money, I think. We can also make those using yeast or bacteria. So, now, here's for the chemists, right? So, non-chemists in the room, you can switch off for a second. Basically, this is just to say that we can do stuff better than the inorganic chemists. So, the bottom of it, where it goes down and back up, they have to use a really expensive rhodium catalyst, they have to use a really expensive ligand, they have to use 50 atmospheres of hydrogen gas, and they get this number 95% EE. We can do this with the transaminase, PLP, which is pyridoxal phosphate, and some other little bits and bobs in there, and this enzyme transaminase can do it 99.95% EE, which is better, lower temperatures, no expensive catalysts, all the products are biodegradable afterwards. Basically, we can do better. So, take that in organic chemistry. So, yeah, what do we want to do? So my group actually works with a catalyst itself called the ribosome, which is inside every living cell, and this is responsible for producing all the proteins and enzymes, or not all of them, but most of them, so should be correct. And what happens is the DNA is encoded into RNA, and this is then read through by the ribosome. And this ribosome then reads this code and assembles the proteins and enzymes from that. And it can do hundreds of couplings a second, whereas we can do maybe one coupling an hour at best. So this is way better than we can do in the lab. The only limitation with this is it can use about 20 different amino acids, 22 if you're being absolutely precise, but I'm just going to stick to the easy stuff first. So it can do 20 amino acids. I will add some new lenses to this. So each lens is encoding an amino acid. I will add some random lenses in there. And these are representing synthetic amino acids, completely random ones that I've made or my colleagues made, that have got completely different chemistry, completely different functionalities to all the naturally occurring amino acids. And so from this, we're going to then take the cells, so we take yeast or bacteria cells, feed them in some sugars, and maybe some genetic information explaining, okay, this is how we're going to incorporate Paddy's special amino acid. And out of that, we're going to then produce a novel enzyme. So this will have novel characteristics, meaning it's more stable, or it works quicker, it can stand acid, or even more unstable, so we want to get rid of it easier. So, for example, diseases which are concerned with proteins which are too stable, we can destabilize them. So, yeah, my idea of the future then is being able to say in every household or every doctor's practice, you'd have some kind of flow reactor or kind of a culture machine where you put in a bit of your yogurt, your sample with your real basic cells, and these would have minimal information there, literally just blanks, ready to go. You can put anything in you want. Then you feed them with some sugars, some trace elements, nitrogen source, real basic stuff, and then you program them with some kind of information. So whether it be a genetic-based stuff or completely man-made polymer stuff, also will be available as an information transfer. And then we want the cells to produce things like insulin or new cleaning enzymes or, who knows, synthetic blood, anything. So we want these cells to produce all these different enzymes.
So yeah, I liken this to 3D printing. So the cells would then produce what we want. So you can make really nice, beautiful things like a piece of artwork or copy the famous artists. Or you can do things like produce it in science where it's normally accessible. So these are some examples of things that have created on the ISS, the International Space Station. And they've done this rather than... Okay, seems to have swapped this out. Okay, so I think I'll put in time anyway. Nearly done. You'll be able to know. So yeah, these are produced on the ISS. And obviously it's better to produce it at the site where it's needed rather than produce it and then ship it elsewhere. This could be applied to medicine. This could be applied to medicine and other production things where it's not viable to produce something and it would be great on the way. Or not viable to send it there. However, this technology could also be exploited. So like the 3D printers, people produce things like guns. So you can see single-use gun or the highlight part on the left-hand structure. That's the only part in America that you actually need to have a serial number for. So people proving you can actually print this out, have an unregistered gun, all the other parts you can buy online, you don't need a license to print it. So yeah, obviously not ideal. And yeah, we could also use this technology as a Breaking Bad reference. You knew it was all coming today, here it is. So yeah, we can also use this technology to make illicit drugs. So it could be used to make things like LSD, THC, amphetamines, but it could also be used to make other therapeutics, which are really, really important for people. So obviously, with great power comes great responsibility. So we have to control this technology carefully, of course. But yeah, the plus side is we come up with a whole new technology. So synthetic spider silk or bone glue, stuff which is then biodegradable in the body, novel materials for body armor. Who knows, new catalysts, so we can do water splitting for fuel cells. We make organic semiconductors or even flexible polymers for OLEDs, polymers which are compatible with skin. We even make synthetic meat and synthetic fuels, so vegetarians in the audience, you have to know, with this technology we'll be able to develop meat which is not coming from cows. Drop all animal husbandry and everyone will be happy. Solve a lot of the global warming problems. We also have endless food supplies. So this is why as an organic chemist I'm a bit of a convert. I just want to say thank you for listening, thank you for your time. Thanks a lot, Paddy.